Namaste, Khushro Vidya. I, JP and Dr. Chandrakala, Assistant Professor, Jyoti Vidyapee, Women's University, Jaipur. In today's session, we will be going to discuss from the course Fundamental of Microbiology. And today's topic for this session is fermentation. Okay, so let's discuss about the fermentation. Fermentation is a metabolism process or a metabolic process that converts sugar to acids, gas or alcohols. Okay, the process of conversion of sugar to acid, gas or alcohol. It occurred in yeast and bacteria. Okay, it is also in oxygen starved or deficient muscle cells, as in case of lactic acid fermentation. Okay, fermentation is a chemical process by which molecules such as glucose are break down and aerobically. Okay. So, in broadly, fermentation is the foaming or like foaming that occurs during the manufacturing of wines, beers, or a process at least 10,000-year old, we can say. Okay. A French chemist or a microbiologist, Louis Pasteur, in 19th century used this technology as in a narrow sense, fermentation describes the change about yeast and other microorganisms growing in the absence of air anaerobically. Okay, and he also recognized that ethyl alcohol and carbon dioxide are not only product of fermentation. Okay, so it is like the definition. If you say fermentation is the chemical transformation of organic substance into simpler compounds by the action of enzymes, complex organic catalysts, and which can produced by microorganisms such as molds, yeast, bacteria, okay, or enzyme act by hydrolysis. Okay, so it's like hydrolysis, a process of breakdown or pre-digesting complex organic molecules to form smaller molecules. Okay, so sugar plus yeast get fermented, and after the fermentation, alcohol and carbon dioxide is produced. Okay. So in 1850s and 60s, Louis Pasteur became the first person like who worked on the field of fermentation and demonstrated fermentation. Okay, caused by the living cell. The first solid evidence was the living nature of the yeast appearance between 1837 to 38, and there was three publications, okay, which proves that microscopic investigation that yeast was living organism that reproduced by birding, okay, and the word yeast should be noted traced in its origin back to the Sanskrit word means boiling. Okay, it was perhaps because wine, beer, and bread was each basic food in Europe, means different European countries. Okay, at that time. So the bacteria was also discovered, and the term was first used by English in the late 1840s. Okay, giving rise to the new germ theory of diseases. Okay. So he defined in many cases, like it was defined like fermentation as life without air. Okay. So in 1877 era, the modern medical bacteriology begins when Koch, a German physician, and Pasteur showed that anthrax bacillus caused the infectious disease anthrax. The epic discovery in 1880 to Pasteur Journal to Germ Theory of Infectious Diseases. Okay. So based on the respiration, like it can be aerobic fermentation, where the oxygen is present, like wine, beer, acetic acid, need of oxygen in the primary first stage of fermentation. When creating acetic vinegar, for example, exposing the surface of the vinegar to as much as oxygen as possible. Okay. Anaerobic fermentation, where the air is absent or in the absence of oxygen. When uh, this method is used, where the energy is extracted from carbohydrates and oxygen and other electrons, etc., are not available in the surrounding environment. Okay. The process can follow glycolysis as the next step, breakdown to glucose and other sugar produced adenosine triphosphate that create energy source to the cells. Okay. And though in this method, a cell is able to regenerate nicotinamide, adenine dinucleotide, okay, NAD plus from the reduced from the nicotinamide adenine dinucleotide, NADH, okay, a molecule necessary to continue glycolysis. So anaerobic fermentation rely on enzyme to add a phosphate group to an individual adenosine triphosphate diphosphate ADP, okay, molecules to produce ATP, which means it is a form of substrate level phosphorylation. Okay. In this contrast with oxidative phosphorylation, which is used in energy for an established protein gradient to produce an ATP. So there are two major types, like anaerobic fermentation can be ethanol fermentation and lactic acid fermentation. Both restore NAD plus to allow a cell to continue generating ATP through glycolysis. Okay, so ethanol fermentation converts two pyruvate molecules, the product of glycolysis, and two molecules of ethanol and two molecules of carbon dioxide. Okay. So the reaction is a two-step process in which pyruvate is converted to acetaldehyde and carbon dioxide. First by the enzyme pyruvate decarboxylase. Okay. So the yeast and the certain bacteria perform ethanol fermentation, where pyruvate from glucose metabolism is break down into ethanol and carbon dioxide. The net chemical equation for the production of ethanol from glucose is like glucose converted to ethanol plus carbon dioxide. Okay. This fermentation process is used for the production of beer, wine, bread. Okay. So it's worth nothing that fermentation in this in the presence of high level of packing results in the production of small amount of methanol, and which is toxic when consumed. Okay. So lactic acid fermentation is a biological process by which glucose and other six carbon sugar are converted into cellular energy and the metabolite lactate. Okay. So the pyruvate molecules from the glucose metabolism may be fermented into lactic acid, and the lactic acid fermentation is used to convert the lactose into lactic acid in the yogurt production. Okay. So it can also be occur in animal muscles when the tissue required energy as a faster rate and the oxygen can be supplied. Okay. So yogurt is made by fermenting milk. It is a high protein, calcium and probiotic. So okay. So like homolactic fermentation, in which only lactic acid is produced and there is no side product is formed. Okay. Then heterolactic fermentation is the in which the lactic acid is produced along with some byproduct like gases. Okay. So the mechanism of the fermentation takes place when the electron transport chain is uh, unusable and become the cell primary means by ATP production. Okay, it turns NADPH and pyruvate produced in the glycolysis into NAD plus and organic molecules. Okay, in the presence of oxygen, NADH and pyruvates are used to generate ATP in respiration, and this is called as oxidative phosphorylation, and it generates more ATP than glycolysis alone. Okay. So first step is common for all fermentation pathway like glucose plus NAD plus ADP plus phosphor, then which give rise to like conversion. The pyruvate is converted to or inorganic phosphate. Okay, ATP and water molecules. So the fermentation is like it is also formed in the stomach of animals and human also. Okay, this process can be the product of fermentation like wines, beers, lactic acid, vinegar, yogurt, cheese. As glucose converted to pyruvic acid, and in the presence of the organism, NADPH is converted into NAD plus. Okay, in the presence of organisms depend upon like Saccharomyces present, so it converts the pyruvate into carbon dioxide and ethanol, acetone or isopropanol. Okay, so it depends upon the microorganisms which converts the products. Okay, so the fermented food are the food that has been prepared in a way so that the bacteria naturally found within them start to ferment. Okay. The process of fermentation is a natural preservative, which means the fermented food can last for long time. Okay. So advantage and disadvantages also there. Advantage is the of fermentation are that lactic acid can be produced and it can produce energy for ATPs. Disadvantage are the production can be slow and the product is impure and need to have further treatment, and the production carries high cost and more energy. Fermentation can provide a rapid burst of ATP in the muscle cells, and even oxygen is in limited supply. However, lactic acid is toxic to cell lactate some extent. So initially blood carries away the lactate. As it forms eventually, lactate build up the lowering of the cell pH and cause the muscles to fatigue. So oxygen so deprived will be there in liver and must convert lactate to pyruvate. Okay. So importance of fermentation is important to cells that don't have oxygen and cells that do not use oxygen. Okay. It allows the cells to convert into two ATPs again from one molecule of glucose without oxygen. Okay. So the fermentation takes away the end product of glycolysis, so the glycolysis can continue. Okay. So it is important in the bakery industries because it is the process that is used the product the bubbles are of carbon dioxide that make the dough to rise. Okay, thank you. Namaste. This session is powered by digital version 2.0 Jyoti Vidyapee Women's University. Hope you are satisfied with my digital session. If you have any doubt, please comment on the inbox or a comment box. I will resolve it, uh, and your problem will be solved in the live problem solving session. Thank you.